This book, Codebreaker by Walter Isaacson, is literally about Jennifer Doudna, gene editing, and the future of the human race as expressed in the subtitle. The discovery of DNA by Franklin, Wilkins, Crick, and Watson in 1950s brought about a revolution in biology, and the book The Double Helix, written by Watson, sparked a feeling in young Jennifer Doudna which eventually led her into the path of the scientist. When Doudna went to Harvard University, Professor Jack Szostak had just decided to shift his lab's attention to RNA when everybody else was occupied with DNA. He suggested RNA as the first focus of Doudna's PhD dissertation, and her journey with RNA began. Their research is to show how RNA can replicate itself. Eventually, they were able to engineer a ribozyme that could splice together a copy of itself, which demonstrates the feasibility of RNA-catalyzed RNA replication. After she moved to Yale, her study was on the RNA structure, and this led her to a field that would become unexpectedly relevant in her career: viruses. She was interested in how the RNA in some viruses allowed them to hijack the protein-making machinery of cells. In other words, RNA interference. Normally, the genes encoded by the DNA in cells dispatch messenger RNAs to direct the building of a protein. RNA interference finds a way to mess with these mRNAs, and at this point, she was introduced into the world of CRISPR. Now, let's see some brief history of CRISPR. In 1986, Yoshizumi Ishino, a student at Osaka, found 5 segments of DNA in E. coli that were identical to each other. These repeated were sprinkled between normal-looking sequences of DNA, which he called spacers. In 1995, Francisco Mojica at the University of Alicante also spotted 14 identical palindromic DNA sequences that were repeated at regular intervals in archaea, and in 2001, he coined the term CRISPR, clustered, regularly interspaced short palindromic repeats. In 2002, Ruud Janssen of Utrecht University in the Netherlands reported the repeated sequences were flanked by one of genes that seemed to be associated with CRISPRs, which encoded directions for making an enzyme. He named these CRISPR-associated or Cas enzymes. In 2003, Mojica found that the spacer segments of a bacteria matched sequences of those viruses that attacked that bacteria. Mojica found that bacteria with CRISPR spacer sequences seemed to be immune from infection by a virus that had the same sequence. CRISPR turned out, indeed, an immune system that bacteria adapt whenever they get attacked by a new type of virus. In 2006, Eugene Koonin at the U.S. National Center for Biotechnology Information extended Mojica's theory by showing that the role of the CRISPR-associated enzymes was to grab bits of DNA out of the attacking viruses and insert them into the bacteria's own DNA. But he got one thing wrong. They and others speculated that the CRISPR defense system worked through RNA interference. That's why Jennifer Doudna, now at Berkeley, would end up getting involved to figure out CRISPR. In 2009, French microbiologist Emmanuel Charpentier discovered that the CRISPR-Cas9 system accomplished its viral defense mission using only 3 components: tracer RNA, CRISPR RNA, and the Cas9 enzyme. The tracer RNA took long strands of RNA and processed them into the small CRISPR RNA that were targeted at specific sequences in an attacking virus. In a conference in Puerto Rico in the spring of 2011, Doudna had a chance meeting with Charpentier, which eventually led to their collaboration. Doudna's collaboration with Charpentier had produced 2 significant advances. The first was the discovery that the tracer RNA played an essential role not just in creating the CRISPR RNA guide, but holding it together with a Cas9 enzyme, and binding it all to the target DNA for the cutting process. The second was the invention of a way to fuse these 2 RNAs into a single guide RNA. It was obvious that this single guide would make CRISPR-Cas9 an even more versatile, easy to use, and reprogrammable tool for gene editing. It was an actual human-made invention, not merely a discovery of a natural phenomenon. Now, the new phase of the research was on a question. If the CRISPR systems will work in cells that have a nucleus, especially multicellular organisms such as plant, animals, and humans. CRISPR worked in bacteria and archaea, which are single-cell organisms that have no nucleus. The Doudna Charpentier paper in 2012 set off a furious competition in many labs around the world, primarily centered on three players: Feng Zhang of the Broad Institute of MIT and Harvard, George Church of Harvard, and Doudna at UC Berkeley. In 2020, Zhang was entitled to a patent for being the first to show how it would work in human cells. But this battle was prolonged due to an unnecessary extent, not because of scientific reasons, but rather because of emotional resentment. This fierce competition could be easily expected when we think about the potential application of CRISPR in human cells. For sure, the era of DNA editing via CRISPR began. The most common and welcome use of CRISPR is in which CRISPR is used to edit some, but not all, of the patient's body cells and make changes that will not be inherited. Sickle cell anemia is one of the best candidates for ex vivo gene editing. In addition to treating blood disorder, CRISPR has been used to fight cancer. Another use of CRISPR editing was to cure a form of congenital blindness performed in vivo. Nature never stops amazing us. It should have not surprised us to see the virus develop anti-CRISPR to fight against the bacteria's immune system over the millions of evolution. Anti-CRISPR was discovered in late 2012 by a doctoral student at the University of Toronto, Giovanni Donami. He attempted to infect some bacteria with a virus that should have been defeated by the bacteria's CRISPR system. In a handful of cases, the attacking viruses survived. It turned out that the viruses had been able to infiltrate the bacteria's DNA with a little sequence that sabotaged their CRISPR system. In 2016, the U.S. Director of National Intelligence issued the agency's annual worldwide threat assessment, and it included genome editing as a potential weapon of mass destruction. As a result, the Defense Advanced Research Project Agency launched a program to support ways to defend against genetically engineered weapons by developing anti-CRISPR to nullify the gene editing. In 2018, a news from China shook the world not because it was a scientific breakthrough, but because it was such an audacious show off of human hubris. He Jiankui, a Chinese biophysicist, created the first human genetically edited babies, supposedly HIV-free. He Jiankui's process involved taking sperm from the father, washing the cells to rid them of the HIV virus, and then injecting the sperm into the mother's eggs. This was probably enough to ensure that the resulting fertilized eggs were free of HIV. But his goal was to guarantee that the children would never later be infected. So he injected the fertilized eggs with CRISPR-Cas9 that targeted the CCR5 gene. The primary concern germline editing is that those changes are done in the DNA of human eggs or sperm of early-stage embryos, so that every cell in the resulting children and all of their descendants will carry the edited trait. Basically, what he did was planting a new human species with artificially edited genes among us. The issue is one of the most profound we humans have ever faced. For the first time in the evolution of life on this planet, a species has developed the capacity to edit its own genetic makeup. In the upcoming decades, as we gain more power to hack our own evolution, we will have to wrestle with deep moral and spiritual questions. Will it be individual choice or community decision to have gene-edited children? Or will it induce free market eugenics so that rich can improve their descendants' genetic pools to make it better? If we're using gene editing for medical reasons, what is the dividing line between treatment and enhancement? After all, are we, the mortals, playing God? In 2020, when the coronavirus pandemic spread across the globe, labs like Doudna's and Zhang's immediately began working on CRISPR-based diagnostic tests. When such tests are widely available, they can be a game changer in the early detection of viruses, as well as the production of CRISPR-based antiviral therapies and vaccines. Unlike the Doudna-Zhang competition for the patents, the one involving diagnostic companies was not very contentious. Both sides know that the technologies had enormous potential to do good. The fact that I'm not that interested in biology seems to be irrelevant when it comes to the question about our future with the gene editing tools at hand. This book is like a miniature of a history of science. It started with the pure pursuit of understanding the mystery hidden in the genes. DNA and RNA, and how life on Earth developed their own makeup to continue to exist over the millions of years of evolution. And there comes the possibility of huge financial gains, and the struggle to become the first and fight over the patent made the whole scene murky. And of course, Dr. Frankenstein always exists among us, and somebody just had to be the first to audaciously create the edited life form. We will not be excused if we neglect to think about this fact that we have tools that our current humanity cannot handle.